In this video, we want to talk about killer T or CD8 T cell activation. And we're going to find that it's a little bit different than the helper T cell activation that requires antigen presentation using MHC2 proteins by antigen presenting cells. Conversely, killer T cell activation occurs in response to virally infected cells or cancerous cells that are producing abnormal proteins, right? Viral proteins or cancerous proteins. And our cells take samples of the proteins we make and express them on the surface using MHC1, right? This allows us to detect abnormalities in all of our native cells. Now, the dendritic cells are once again going to respond to these foreign antigens by phagocytosing them and presenting them using MHC1 proteins, right? In terms of activating helper T cells, we required MHC2, right? So the dendritic cells actually present foreign antigens using both. Now, once again, those dendritic cells will, upon phagocytosis, will migrate into the lymphatic system where we have our pool of naive B and T cells. Right? And this dendritic cell is going to present that foreign antigen using the MHC1 proteins to the CD8, right? The naive CD8 cells. And when they find a receptor match to this foreign antigen, we're going to get activation. Now, in many cases, we actually require helper T cell activation of this CD8 cell as well. In other words, the helper T cell will recognize the same antigen on the MHC2 complex. It becomes activated, starts to secrete interleukin 2, and helps to activate this CD8 T cell. Right? So we get this sort of dual stimulation. And that's really important because CD8 T cells, unlike the helper T cells, actually will kill cells. Right? We want to be very careful about activating them. The helper T cells help. Right? They don't directly kill other cells. Now, upon activation, we do get that clonal selection and expansion, right? That's that process of division, cell division, and differentiation. We get our memory CD8 T cells for a secondary infection. And we get our large pool of effector CD8 killer T cells. Now, these will migrate out of the lymphatic system into the interstitium, into the bloodstream. They get drawn out chemotactically at sites of infection and inflammation. And those activated CD8 T cells will recognize foreign antigens now, right? That same foreign antigen. And once they contact those infected cells or those cancerous cells, they release a series of cytotoxic proteins that will create pores in the membrane and also initiate apoptosis, causing those virally infected or cancerous cells to die.